Dear attendees, I am very happy to have you online to discuss about something which is quite important, which is the population bioequivalence and the average bioequivalence also for the inhaled products. So that is the webinar of today, which is made using Certara software Phoenix with Nonlin to make all the calculations. So the first thing that we are going to start with is a very simple introduction. So what we have to take into account is that when we are going to calculate this type of metrics, it is for locally applied, locally acting product. And what is a locally applied, locally acting product? It's a product which is by definition applied locally, and that is going to have its effect on the site of application. If you have any systemic action, it will be seen as a side effect and something which is not desired. For example, among the locally applied, locally acting drugs, you have the dermatological products, the eye drops, the ear drops, the nasal products, the orally, vaginally, rectally applied product with act locally, and also the inhalatory products. And what we are going to discuss today, it's more about everything which is going to be inhaled product or nasal products. So for the locally applied, locally acting drugs, we are going to have very often the possibility to have bridge applications, mainly for generic, for example. What are the duties and the pre-requirements in this case? You must have a therapeutic equivalence between the product and compared to the product which is already approved, that means the reference listed drug, with regard to the efficacy and safety. So that the data which are going to be used for the innovator can be bridged with your own data. The bioequivalence data are not a suitable way because very often you cannot measure any plasma levels. And in this case, you may, if you cannot make any bioequivalence study, make clinical trials. But as the clinical trials are very heavy, you could, in some cases, use other type of models, for example, in vitro models, to demonstrate the equivalence between the formulations based on some quality attributes which could be linked with the therapeutic activity. So you have a lot of tests for nasal and inhaled product. Among all those tests, you have, of course, the delivered dose. But you have also the drug substance particle size distribution, the droplet size distribution, the single actuation content through the container life, the aerodynamic droplet size distribution by cascade impactor, the priming and repriming, the mean delivered dose, the plume geometry, and so on. You can see that when I want to show the equivalence between two products, you may have to use more than one matrix. But the problem also later on is how to analyze all those data. So for the analysis of data, you have more than one option, which is the existing. Of course, at the beginning, you can make only descriptive statistics. But descriptive statistics, it's a very simple approach, which is not pertinent, as not associated with any risk, and mainly not the alpha risk, that means the patient risk, and you cannot control it. So you have two other types of statistics that you can use. You can use something which is the average bioequivalence, or ABE, or the population bioequivalence, PBE. You have two approaches, mainly for a very simple reason, is that PBE is more focused for the US FDA, and ABE is more focused for the European authorities. We are going, in the present case, to be based on the draft guideline of budesonide, which was published by the US FDA. Why we are going to use this guideline? Because they publish inside this guideline some data that you can use to validate your software. So the basic is going to be this draft guideline. As I told you before, you have a difference of approach between EMA and FDA. What is the difference between the two? The difference is that ABA has fixed limits, and those fixed limits which are described is 0.9111. PBA, in contrast, has limits which are function of the reference variability. That means you will have enlarged limits when the reference variability is going to be large. Now we are going to focus on the PBE. PBE, first of all, the introduction. So you have a very nice paper which was published in 2014 by Morgan. This is the reference which is here. So what is PBE? PBE is a metric which represents a combination of difference in mean and variance, scaled to the reference product variability or variance for transformed data. The transformation used is natural log scale. So what is the interest of this type of thing is that you try, in this case, to have all the distribution which is going to be focused on a single matrix. And this matrix is going, in this case, to be compared to some limits. And if this metric is below the limit, in this case, is going to be accepted. The equivalence is met if this function of the population parameters is less than an user acceptance limits, which is theta BE, or very often as a Xi parameters, which is the linearized criterion, which must be less than 0. The approach which is described in the budesonide guideline is this later one, that means the linearized criterion. So the first thing that we need to have in mind is that we are going to work on log transform data. That it's based on the fact that we assume that we have a log normal distribution and that later on we can use multiplicative models. We are going to use a 90% or a 95% confidence interval. In the present case, it's going to be a 90% confidence interval. And in this case, the upper bound of your 90% confidence interval must be below a fixed limits. We are going to take into consideration the variability of the reference compared to some constants which are prefixed. And you will see that the constant which is given in the guideline is 0.1. This 0.1 means nothing more than the log of 111. In case of large variability of reference, the acceptance criteria are going to be relaxed. So what is said? What we have here in the draft guidance for budesonide is this type of formula. You can see that is something which seems not to be so easy to calculate. But later on, we have also other things which are in this guideline. And what we need in practice to calculate everything, it's first of all a stepwise approach with these parameters. The first one, which is delta, is the difference between the two means, the mean of the test and reference. Why we have a difference? Because we have a log transformation. And you know that log of t minus log of r means is the log of t divided by r, it's a ratio. We have m, which is the number of life stage. That means beginning, middle, and end of the life stage. Then we have n, which is the number of vials, bottle, or canister for per batch, for test or reference. L is the number of batch, test or reference. Then you have mean sum of square. You have two of them, MSWT, which is the within bottle variability of the test product, MSWR, which is the within bottle variability of the reference product. Then you have MSB T or R minus MSW, the previous one, T or R divided by M, which is nothing more than the between bottle variability either for the test or reference product. Then you have a degree of freedom, which are going to be associated with all this mean square, and you have a T and a chi-square distribution. So what you need at the end, it's a very low number of parameters. Is that a problem when you see the guideline? A little bit, because you can be panicked. You can see that the MSB and MSW are sum of value and differences and square differences. Then you have square root of this mean square. Then you have a lot of other things that you have to calculate using chi-square distribution or T distribution. But do not panic. At the end, it's not so complicated to calculate everything. So the first thing that you have to keep in mind is that the mean square is nothing more than something which you could extract from the analysis of variance. In Phoenix, you have some analysis of variance, which is a mixed linear model, for example, that you can use. In this case, you will have the parameters which are here. The mean value for the test and reference, you can have it with the descriptive statistics. So a part of what I just showed before, that means this one and this one can be extracted from the ANOVA, and this one can be extracted from the descriptive statistics. So you can see that it starts to be a little bit more simple. So what do I need? I need also, you can see here, something which is a t-distribution, and here a chi-square distribution. The problem is that in Phoenix, you don't have a t-distribution. That means you need to have a table with a t-distribution. For the chi-square distribution, you have a function in Data Wizard which allows you to calculate the chi-square distribution. If you prefer, you can create the chi-square distribution table at the same time as a t-table. It's up to you. And the rest, if you have a look, is nothing more than multiplication, subtraction, division, and also square root that you are going to use. So that means it's not very complicated mathematics. So in Phoenix, you have some function that you can use here for the calculation, but also ANOVA and descriptive statistics. So you have a lot of things that you can do here directly in Phoenix without any further calculation by yourself. Something which is quite important to remember is that Phoenix is working within line for calculation and not between rows. That means you have, in this case, to put everything on a single line to be able to make all the calculation. That means that you may have to merge or to join a lot of files together. For that, you need to have a key which allows you to make the merge of everything. In practice, how can we make the PBE calculation? You will see later at the end of this talk that Mouli is going to present a template. I am going to present another template here, which was made more to explain you step by step all the calculation. The first thing that you have to do is to prepare the data. Preparing the data is a very important thing that you have to do. What we use the current example of budesonide. But we have to take into account that within this example, the data were already log transformed. So what I have made, I have transformed back from the log to the original scale, such as you will have the data when you make your own experiment. Then I will do a step-by-step programming, and I will try to explain you what I have done. So what do you have to do? The first thing that you need to have is a data set with the original data, with batch, container, stage, product, and the data. In this case, I have transformed back all the data of the budesonide guideline in the original scale. You need also to have something here, which is going to be a t-table and chi-square table. The chi-square, you have a function in the data wizard, which is chi-inversed with p and degree of freedom, but I have created here my own table. You can create that using Excel or importing statistical table for any website. You can see that you have something here which is flag. That is something that I'm going to use to merge my files together. I also add in my data step something which is the constant table, in which you have the higher limit and the sigma tau, which is the constant value given by the FDA. If I have my original data which are here, I can transform them in log in a data wizard using a function which is the log function, of course. So the first thing that you have to do is to prepare the data. And when you have this table about the t-distribution and the chi-square distribution, you must not alter it, otherwise it's not going to work anymore. The same thing for the constant which are here. And in the templates that Mouli is going to present, you have exactly the same thing presented in another way. In what you are going to have at the end of these lectures is the possibility to have both Mouli's template and my template that you can download. You have to prepare the calculation. What we have to see is that the calculations here are going to be, first of all, the mean square, which are here, and the mean values. Then I have to calculate the mean square divided by m and those values here for the h and then the u. That means the e, the h, and the u. So what we are going to see now is how we can calculate everything here. So to prepare the analysis, the first thing that we are going to do is the mean value. So after you have transformed the data in log, you are going to use the descriptive statistics to calculate the mean. And that is the mean value for the test and reference for all the data. You can see that you have 90 measurements for the test and 90 measurements for the reference, which is M. Then you have the mean value, which are here. You are going to use the linear mixed model to make your ANOVA, which is going to return the mean square MSB, MSW, and so on. What are you going to do? You are going, in this case, either to use the unchanged and untransformed in log data, and in this case, you must use here a transformation in your linear mixed model, or you use directly the log one, and in this case, you do not make any transformation here. What is important is to sort by product, that means test and reference, and to use the container and only the container as a classification here. So you will have a very simple effect, which is going to be a fixed effect with container, nothing more. Based on that, what are you going to do? You are going to have and to return something which is going to be, in this case, those values in the partial sum of square for your ANOVA. We have seen before that you had the mean, and now that you are going to have here is on the log data, you are going to have the container and the error, degree of freedom, and mean square. But I need to have it for the test and the reference in two separate files. And I need to have the container and the errors. That means I have to split this one in 4 subsets. And these subsets are going to be split by product and hypothesis, the hypothesis being the container and the error. So I am going to have at the end 4 files. One for reference container, second for reference error, third for test container, fourth for test error. When I am going to have that, I'm going to join them. I am going to join all these single, very small files to have everything in the same line for the test, and I will make the same thing for the reference. That means I'm going to merge, in this case, my container and my error files for the reference, and I'm going later on to make the same thing with the mean value. So I'm going to use to join them a key, which is going to be the product. And I am going to have the degree of freedom, the mean square 1, the degree of freedom 2, the mean square 2, which is going to be, in this case, the containers and the error term, and here the mean values. So that means I start to create everything. Based on that, what I'm going to do is to rename everything and to make my calculation to have my M, my N, and my L. It's very important when you do it to have a name which is going to be easy to be connected with what is in the budesonide guideline. So that means you are going to rename some of them and to make extra calculations. As you are going to have a template, I'm not going to go in detail in the calculations, but only to show you, you have the rename which is here. DF1 is going to be DFB, that means degree of freedom of B. MS1 is going to become MSB, and so on. Later on, you make the calculation, and you are going to make the calculation to calculate M, and the M is nothing more than the total number divided by the degree of freedom of B R plus 1, and that is directly calculated. I'm going to include here also something which is very important, which is my flag equal to 1, which is going to be my key to merge everything together later on. Then I am going to make the same thing for the test. And I am going to have at the end something which is going to be the join of the test and reference, and I'm going to have a huge line like this one. In this line, I will have my mean sum of square, which are going to be here, my M, N, and L, as well as the mean values, my constant limit sigma tau, which is here, and I will have the same thing for the test and reference. Based on that, I can make my calculations. And inside my calculations, I will be able to calculate, in this case, all the value that I need. That means the age and so on. When I have done it, now I need to continue to make my calculations to include my t-value and chi-square values. It's a huge file like this one with my flag to be allowed to merge it later on. And I will have at the end nothing more but my chi-square, my degree of freedom, and my value, which are here, which is the n, which allows me to know what is going to be the chi-square of the t distribution. I'm going in this case to say something quite simple, is that when I want to have it, I'm going to compare 58 here with a number, and if it's the right number, I'm going to select the value that I need. 1, 67 here, 15, 52, 8. You can see that in this case, I'm going to return all the cells which correspond to what I need are going to be full of a number, and the other are going to be blank. So I need now to, as before, to have everything on the same line to be able to merge everything. How can I do it? I can do it on a very simple way. I cannot remove the blank cells using Data Wizard because very often in this case, either I will move the line or I will have still all the lines. So what I'm going to use something which is a very simple thing, to use descriptive statistics. As I have only one value by column, if I make my descriptive statistic by columns, that can be the mean, that can be the sum, that can be the minimum or maximum, I will have the right number. So I will have at the end something like this. And I will have something like this only because I'm going to pivot my results to have it everything in line. And of course, I will keep my flag being here to help me to merge later on with my previous data set. When I am going to merge it, I am going to have something like this, a huge line with all my values, including, of course, the chi-square and the t. As I told you before, you do not need to have a table with a chi-square because in the Data Wizard you have chi-inverse that you can use directly. So now I have all the values which help me to calculate H, E, and U. That means I can calculate everything. But you can see that you have two ways to calculate it. You have ED, E1, E2, the same thing for the two upper and bottom table, but then you have E3S, E4S, or E3C, E4C, which means that you have either a constant value or a scaled one. That means you are able to calculate both of them. So as it's more easy to calculate everything and later on to give the options to know what you are going to use, I take the options to calculate both of them. That means the scale and a scale matrix. To calculate it, it's not complicated. You have a lot of steps you can see, but it's only addition, subtraction, square root, and so on. So you can see that it's not complicated mathematics, but the risk that you have here is to make a mistake. So it's very important, starting from the beginning, to have a system of notations which is going to be very obvious in such a way that you can be sure that you are not going to make any mistake. So you have a lot of steps and you have risk to make mistakes, so you have to be careful, and it's more important in this case to have a template which is validated to be sure that you are going to make always the same type of calculations. So when you have it, you can calculate all your metrics. What I have done here, I have done something which is both calculations with its reference scale and not scaled. And then depending on the value that you have, what I have, condition fulfill yes or no. That means the non-scaled is not fulfilled and the scaled one is fulfilled in this case. And I have the result which is going to appear only when the condition is fulfilled, which is the reference scaled one. And the result is going, I am bioequivalent in this case. So it's a lot of calculations, but it's not something which is very complicated. When you have made all your calculation, you need now to present them. And it's quite important to have a way to present your data which is going to illustrate everything. Among the presentation, something which is quite important is to make a box plot. The box plot is going to give you the dispersion of your data as well as their mean or central values. You know that a box plot is nothing more than a representation of all the data in one box with whiskers. In this case, I have my 3 reference batches, my 3 test batches which are here. For each of them, I have a box and whiskers. My box corresponds to the first quartile and to the third quartile. That means Q1 and Q3. That means within the box, I have 50% of the value. And outside of the box, I have 25% here and 25% here. Then I have whiskers, and the whiskers has a length which is going to depend about the variability of your data. You can have, of course, some points which are here, which is the, into quote, outliers. That means the extreme points. What you can see here is that somehow the reference drugs has a lower variability. But in some cases, for the reference, you have data which are into quote outliers, even if the drug is correct and with good quality. That means you have extreme data. What we can see here is that for the test formulation batch 4 and 5, you have quite a huge dispersion, which is not the case for the batch number 6. And then you can use the table tool to create something which is a very simple table like this one. What is the higher limits? What is your HN that you are going to use? You know that if in this case the H is going to be below 0, you are bioequivalence. Are you going to be reference scale? In this case, it's yes. And so my result is I am bioequivalent. I am bioequivalent because I have a variability on the reference. This variability is due to my extreme values, which are here. So we can, and what we had made, we have verified the calculation. We have verified the calculation by 3 means. The first of all, you have the Phoenix results, which are here for the reference scale and constant scale. That is a value which are displayed in the FDA guideline, and you can see that we have the same one. Of course, sometimes we don't have the same rounding in number of digits, but we have exactly the same value when you use the right number of digits. What I have made also, I have recalculated everything using Excel, and I have recalculated everything using SAS to be sure that everything is going to work correctly. And you can see that in this case, both Excel, SAS, FDA, and Phoenix return the same results. But that is not a proper calculation and validations, because if it will be a proper validation, in this case, you must also test what is going on if one value is missing, what is going on if you have not the same number of value for test and reference, and so on. That it's only a way to show that the calculation performed based on the example return the same result as those which are published. So what can we say as remarks? We have some remarks which are quite important. What is going on if you have m equal 1? That means if you have only one life stage. You have seen that in the current example, you have 3 life stages, which is beginning, middle of life, and end of life of your containers. But in some cases, you can have only one measurement. If you have one measurement, it's very easy. You are going to ignore 2 of the value, which is E2 and E4, E4S or C, depending if you are scaled or non-scaled, that means constant. And then you are going to calculate everything with E2 and E4 equal to 0 or not existing. But somehow, very often it's going to be rare, because when you have this type of data of nasal product or inhaled product, very often you are going to make it at beginning and end of life at least, and sometime beginning, middle, and end of life. For the data collected from more than one stage, you are going to use the equation which are described just before. So the fact to have m equal to 1, it's not a problem, because that can lead to calculations which are correct. So to go a little bit in detail, when m equal 1, that means the term which is here is going to be equal to 0. And if this term is going to be equal to 0, that means E2 is going to be equal to 0. That means you do not have any intra-bottle or intra-vial within bottle variability, nothing more. What we have in Europe, it's an average bioequivalence calculation. This average bioequivalence calculation, it's another approach, very close to what we do for bioequivalence. So that means we can use something which is very easy, is to use in this case the bioequivalence tool. with parallel design or other type of design, average here, reference formulation is going to be reference, and the dependent variables is going to be the data, and you are going to use a lot transform in this case, and the formulation effect is going to be the product. You are going to calculate a 90% confidence interval, and that is going to return everything directly here. And you can see that in this case, I'm not going to be bioequivalent. That is very close to the constant scale in comparison to the reference scale metrics that we have seen just before. So now I'm going to give the floor to Mouli in 2 seconds about the Phoenix template. Mouli had prepared some templates which are based on Phoenix 8.3 or greater as a version. What is quite important inside this template which was provided, mine was only for teaching purpose and for training to explain you how it's working. This one is more for really to be used. You have a first step, which is the user step, and Mouli is going to describe it perfectly at the end, just after me. And that is going to help you to map the data. That means you must map, in this case, the data that you enter. And in this case, for example, for me, I enter lot vial stage product and in vitro. You have to say that the lot is going to be equal to the batch, the vials to the container, the stage, the product, and in vitro to in vitro measurement. That allows to have something which is going to simplify, to rename the header of the column from lot to batches in such a way that everything can run later on. So now I am going to give the floor to Mouli, if that is okay for you.
Okay. Welcome, everyone. Thanks for giving this opportunity, Professor JM. I'll be taking through, my name is Chandramouli Radhakrishnan. I'm application scientist with Certara, and I'll be presenting on the template that we created. A couple of things we would like to point out that the Phoenix template has been tested by the authors. In this case, it's going to be myself and Anna Henry, and also Professor Jane Michael as well. This particular template did not follow the formal testing and validation procedures for Certara software development. In this presentation, what I'm going to take you through is about the 2 templates. One is PBE ABE version. So this is a project cover, the template name, and this was prepared by Professor JM, and which he had gone through in detail in explaining how the calculation is being done. And I'm not going to revisit those calculations again in both of these templates when I'm showing a demo. The template 2 is what we from Certara started working on it, and we developed this one. And where can I get these templates? So there's a link there to Auvergne University, and it's free to download. Both the templates and the presentation will be made available after this session. What can you achieve with these versions? So you could do a PBE developed based on US FDA guidelines and EMA based analysis. And what is not available is with the template 2, we haven't tested the life stage calculation, which M equals 1. So that is not being tested. And conventional PBE using D10, D50, and D90 and Spanish is not available with the current template, with both the templates, and also EMD, the earth moving distance. So these are for our future work, which we are working on to make this happen. And again, I need to point out that this template has been tested by the authors, and it does not follow Certara's software development validation process. You could use this software for your calculation. We are in the process of making it more precise and make it even more better than what we have at the moment. Having said that, I'm just going to move on to Phoenix. So I've got these 2 templates loaded, as you could see here. The first one is about PBE ABE calculations. So this was prepared by Professor JM, and these are the calculation steps and sub workflows which I will take you through each and every calculations individually. You got these data which are taken from FDA guidelines. and where you have your data and the log distribution functions. And this is something which he showed in his presentation, the comparison where he used Excel, FDA, and Phoenix as well, and he compared the results. So these are stats table which we would recommend not to touch because these are used for your calculations, and it's about the limits that's being used in the calculation. So these two tables, if you need to use these template, I wouldn't recommend you to meddle with these two templates. This is a detailed template that goes on, and I'll be concentrating more on the second template because in this template, just as you mentioned, it's just a guideline for you to go through the process, how the calculation is being done. The second template is what we started developing. When I say we, in this, what we had added extra is about this guidelines documentation. So if you want to look at the guidelines documentation, all you could do is just double click on these, and then it will ask you to load the document in an external viewer. And if you have a PDF attached, so you have your documentation guidelines ready to use. Similarly, we have that one done for some instructions that to go on, what is needed, and the way you need to map it. So this is something which Professor JM covered in his last slides. In this case, as we specified, we have clearly said do not modify. So I would recommend not to change any of these values because these values are used in the calculation process. And as this is again the FDA comparison with Phoenix as well. This is the FDA guidelines data taken from the publications. As you could see here, the batch container stage product data. The template can support both alpha and numerical values as well, or the mixture of both. These templates all red because I've removed the data sets. What I'm going to do is I'm just going to drag and drop my data set. So it's once I dragged and dropped, you could see all the data are being mapped. As mentioned, you can have different names, but this has to match what you see up here. So it can be container, can be, you can name container, as a bottle, or batches can be named as lot. You can have lot here, but lot has to be mapped with a batch, and you can have in this case, it's a product. You can have it as a treatment. So the treatment has to map against product. This is something a must. You need the way it needs to be done. So once this is done, I'm just going to execute this workflow. And the next step is to execute the master workflow. As you could see here, these templates are all interlinked, and it does the calculation for you on a click, and the reports are ready for you to use. You can save this template, and you can execute on a click, and you got your results. So we have used just reporter function. This is basically what you see in the FDA guidelines with some of these recommendations, like where you have your standard deviation and also the summary table ready to go. As mentioned, I also added some box plots and some descriptive statistics and table which you could use in your report. So you have your product reference touched and with all the standard deviation mean ready to go. So as I mentioned earlier, I just once again need to point you out that this is not developed as per Certara's software development guidelines. These are developed by us. And as I mentioned earlier, you have these presentations, these templates available at certarauniversity.com, and we'll be sharing these slides with you, and the link is also available for you to download these templates. If you have any questions, we are here to answer these questions.
Gentlemen, I do not see any questions popping up. We thank you for joining us for this webinar, and this concludes our session.